I'm looking forward to giving you an update on the past year with Island Pharmaceuticals. Thank you for those who are here today. Thank you for those who are here online. If we could have the next slide, please. As I think we're all keenly aware, Island Pharmaceuticals is an antiviral drug development company. We're focused on targeting infectious diseases. We have, over the course of the last year, really fine-tuned our corporate strategy as we look to capitalize on emerging infectious diseases, emerging viruses that perhaps might play a role in biodefense, looking for countermeasures. This is consistent with our theme of developing a small molecule against dengue, as well as some other viruses that we're looking at. The assets that we'd like to look at, we're clearly focused on being a clinical stage company. We want to be in clinical trials, not doing early stage R&D. And as a result, the molecules that we find interesting are those that are small molecules, that have exhibited antiviral activity, that have open INDs with the FDA, may be eligible for priority review vouchers. We think this is a really important part of our strategy. We may have interesting or expedited paths to approval. Let's go to the next slide, please. It's been a very exciting year for Island Pharmaceuticals. We have accomplished a lot and hit many different milestones. Early this year, we were announced that we were the beneficiary of about $625,000 in Department of Defense funding that went to support our phase 2 clinical trial that we're conducting right now. This was a grant that was given to our clinical site, SUNY Upstate in Syracuse, New York. We continue to process patent applications. These are the applications that we've licensed from Monash University. We continue to have success getting these cases allowed, so I'm always happy to see that. Earlier this year, we concluded a dose escalation study. This was a clinical trial that we ran in Australia, and it turned out to be an incredibly important clinical trial for us. It gave us data that helped us fine-tune our strategy and dosing regimen for the phase 2 trial that we're running right now. So that turned out to be very, very useful. In addition, we had a rights offering that we raised $1.95 million in March, again, to support these programs. The data that we obtained from the dose escalation study, it was a single ascending dose escalation study. We were able to use modeling to predict the dose that was going to be useful when we did multiple days of dosing with ISLA 101 in our phase 2 clinical trial. That again turned out to be incredibly useful because it told us this is the dose that we need to use if we want to achieve a particular blood concentration of ISLA 101 that would be effective, hopefully, against dengue infections. In view of the fact that we had really nice data coming out of the dose escalation study, we were able to fine-tune the protocol for the phase 2 clinical trial. Rather than doing a phase 2 dose escalation study, we were able to shift and do a single dose, but looking at both the ability of ISLA 101 to protect against a dengue infection or to treat a dengue infection. This is the study that we're working on right now. So we submitted a revised protocol to the FDA. The FDA was, oh, you know what? I'm just missing. I'm looking up here and I'm missing the last column on my screen. I'm just going to turn and look at this screen if you don't mind for a moment. We filed our single dose escalation study with the US FDA. We then submitted a revised protocol to the FDA, which we had changed the protocol for our phase 2 clinical trial to protect. You may recall that it was called PEACH initially. It's now protect, which includes the ability to both protect and treat a dengue infection. We were very happy to announce that we kicked off the phase 2 dose escalation study. No, I'm sorry, the phase 2 prophylactic study. This was kicked off about 45 days ago. We began dosing these individuals, these healthy subjects in our phase 2 clinical trial. We've also talked about for a while that we're looking at other assets that we want to bring into the company, and we were able to identify an asset that we like a lot. I'll tell you more about it throughout the presentation. The molecule is called galidesivir. Again, we like this very much. We've signed a binding letter of intent for an option to acquire this asset, one that we think is going to be very exciting for the company. I want to thank Dr. David Brooks for his time on the board. Dr. Brooks announced his retirement from the board earlier this year. And I want to welcome Chris Tumanopoulos to the board. Very happy to have you here, Chris.
Thank you.
I want to thank Paul McClayman and Anna Lavelle also, who are retiring from the board. I appreciate their effort And I want to welcome Phil Lynch, who's here with us today. Phil, welcome to the board. We're excited to have you on board. It's going to be a very exciting time over the next year or so. Thanks. Finally, I'll close this slide with the announcement that we raised $3.5 million just a few weeks ago. Really exciting opportunity to get an investment from some high quality, sophisticated investors. We're really excited by the support shown from this group of investors. And this is capital that will enable us to complete the phase 2 clinical trial that we're in right now, as well as execute on the acquisition of galidesivir, a molecule that we all, again, like very much. So again, it's been a pivotal year for the company, a very exciting year, and really sets us up for another exciting year next year. We can go to the next slide, please. So ESL 101 is our lead indication, and I suspect that we all know that ESL 101 is a molecule that we're pursuing against dengue infections. And dengue infections are no joke. It is around half the world's population is at risk of an infection with the dengue virus. There's 4 strains of dengue. There's about 400 million infections a year with the dengue virus. About 100 million of those require some form of medical intervention. The only medical intervention that you can give these individuals is symptomatic. You can't give anybody an antiviral to reduce the effects of a dengue virus. They just don't exist. That's what we're doing with ESL 101. We think there's a huge opportunity. There's a huge need for a molecule like this. Next slide, please. The problem is huge, and the problem is only getting worse. This is a really recent piece of data that we have seen. And what it's showing you is over the course of the last 10 years, the number of dengue infections. And look at 2024, the graph on the far right. By far the worst year on record. It is a huge problem. It's a growing problem. We're seeing dengue infections now, not only in tropical regions. We're seeing dengue, as we'll see shortly, moving into the southern part of Europe, in the United States. It's a growing problem, and again, one for which you really can't do anything right now. There's just no therapeutics for it. Next slide, please. Just to make sure we're all on the same page, just to remind you that dengue is a mosquito-borne virus. You don't get it by breathing on people. You get it when a mosquito bites an infected person and then goes and bites an uninfected person. So the mosquito is the vector that transfers the dengue virus between individuals. And these mosquitoes, they like to feed. A mosquito can ultimately infect 100 or more individuals. So once you have a locally acquired dengue infection and you have mosquitoes there who can be the vector, the numbers of infections can increase exponentially. It can become a huge problem. One of the reasons that I think the military is very interested in what we're doing, a number of people are interested in what we're doing, because you simply can't have people getting sick when they live in close proximity to each other like this. So again, it's a mosquito-borne infection. Next slide, please. Part of the reason we think that we're seeing such a dramatic increase in the number of infections is we typically have thought of one particular mosquito species as being the predominant carrier of the dengue virus. This is Aedes aegypti. This is shown in the red line. What we're looking at in this graph is the incidence of dengue virus infections caused by different mosquito species. And it's showing an increase over the last several years. And you can see, if you look at the red line, this is the incidence caused by Aedes aegypti. And it's increasing at a pretty slow rate, but there's already lots of these mosquitoes out there. But look at the blue trace. This is another mosquito called Aedes albopictus. It's known as the tiger mosquito. Aedes albopictus, I think you can all clearly see, is increasing at a dramatic rate. And the interesting thing about this mosquito is it doesn't necessarily care to live in the tropical regions where Aedes aegypti likes to live. It actually prefers more temperate climates. So this is what's moving into Europe right now. This is the mosquito that is causing significant concern throughout Europe and other parts of the country where it's not typically considered tropical. So if you live in an area where you thought you didn't have anything to worry about, I would caution you to be aware and pay attention to when you see locally acquired dengue infections. Los Angeles, San Diego in the United States, these are not tropical regions. They're experiencing locally acquired dengue outbreaks right now. So as we say here, the train is coming down the track. And currently, there's nothing that we can do about it. We think ESL 101 may be able to curtail that train. Next slide, please. Again, to harp on this, we're reiterating that there's a significant problem. Today, if you were to look at a map of the dengue infections in Australia, you would not find many. Likewise, in the United States, while you would see some in California, you would see some in Florida and some in southern Texas. What we're seeing on this slide is where is dengue predicted to be endemic by the year 2050. Now look at Australia. The northern half of the country is predicted to be endemic. It's going to be a significant problem. The southern third of the United States, southern Europe, all predicted to be endemic for the dengue virus. Again, there's nothing that can be done about it today. Next slide, please. So what are we going to do about it? ISLA 101 is the measure that we're developing that we believe, based on robust preclinical data, could be a game changer for dengue. Next slide, please. ISLA 101, as I suspect many of you know, is a molecule we became very interested in a number of years ago. It's a molecule that actually was developed by a large pharmaceutical company as a cancer drug. As a result of that, it has been studied in many, many clinical trials. It's been observed in thousands of individuals in clinical trials. It's got a well-known safety profile, yet it's never been approved for anything. We became aware of ISLA 101 when we were looking for molecules that had activity against the dengue virus, and found some excellent work at Monash University that they had published showing that ISLA 101 had activity against all 4 strains of dengue, as well as West Nile, Zika, yellow fever. The interesting thing that I like about this as a scientist, this is a very unbiased approach to drug discovery. They screened thousands of molecules looking for a molecule that had the activity they were interested in, and found one. It was ISLA 101. Monash went on to characterize the work, performing mouse studies showing that ISLA 101 protects against infections with a lethal strain of dengue, and I'll show you these data. The other thing that I love about the story is weeks after Monash published their work, a group at Harvard published nearly identical results. Again, screening thousands of compounds, looking at molecules that had anti-dengue activity. They also arrived at ISLA 101. And the fact that 2 world-class institutions perform these independent screens, unbiased screens, arriving at the same molecule, gives us great confidence that what we're seeing is real. Next slide, please. This is an experiment that is really what convinced me that I wanted to spend the next several years of my life working on this program. This is an experiment that Monash University performed. It is an animal experiment, and we are studying a dengue infection in mouse, and this is a lethal strain of dengue. So what you would find is we're looking at the x-axis, time after infection, looking at the y-axis, percent survival, and you can see at day 0, you have 100% survival. This is when you infect these animals. If you look at the red line, you can see this is animals that were infected and not treated, and 100% of the animals are dead, 0% survival. If, however, you give 1 dose a day of ISLA 101, look at the green line here, this gives you about 20% survival of these animals. If you give these animals 2 doses a day of ISLA 101, now you see about 70% survival of these animals. And in the drug development world, when you see dose responses like this, increasing concentrations resulting in increased effects, it gives you confidence that what you're observing is a real effect that's caused by your drug, okay? So we like this very much. It gave us an indication that ISLA 101 is having effect. It is preventing death in this otherwise lethal strain of a dengue infection. Important to note, the dose that is being dosed in these animals right here, we're able to calculate that. We're also, I am able to tell you that the dose that we're using in our current phase 2 clinical trial that we've recently completed dosing is about 10 times the dose that you see at the high dose here. So again, we believe all of this data strongly supports carrying ISLA 101 into the clinic, which obviously we've done, and I'll tell you about shortly. Next slide, please. The phase 2 clinical trial that we're conducting is, I'm very excited about the structure of this study. We're running the study at a site, SUNY Upstate in Syracuse, New York. It is a phase 2 clinical trial in dengue-infected individuals, and it is a challenge study. And what I mean by challenge study is enrolling healthy individuals and then intentionally exposing them to a strain of dengue, an attenuated strain of the dengue virus. These people get a little bit sick. The US Army has run controls with between 15 and 30 people running controls through these phase 1 studies, just looking at the virus. This was a virus that was initially developed as a vaccine. It just happened to make people a little bit too sick to be useful as a vaccine. So rather than get rid of that virus, the Army said, let's run a phase 1 study. Let's see how people respond to the dengue virus. And again, people get a little bit sick. Typically, they have a rash. Sometimes they get fever. Nobody gets very sick. we saw these studies and reached out to the Army and said, what we would like to do is run your study with your virus, but put our drug on board. And they agreed. So we now have a cooperative research and development agreement in place with the US Army. They're giving us the virus that they used. They're giving us all of the data that they have obtained out of their phase 1 study, which creates really a control comparator database for us so that we know what a wild or a dengue infection looks like in this model system. And then in exchange, they want to see our data, which we're happy to show our data to the US Army. We think that would be a really nice collaboration that we would like to continue. Let's go to the next slide, please. So the way the study is designed, we're dividing this into 2 cohorts. The first cohort, which we call the phase 2A cohort, is a prophylactic study, meaning we pre-treat individuals with ISLA 101, then expose them to the virus, and then continue treating them for about 20 days. These individuals come to the hospital every day or every other day after the second week. They see a physician, they are examined, they give blood samples, and all of that is because we want to be able to measure the blood concentration of ISLA 101. We want to be able to measure the virus level in those individuals who have either been exposed to a placebo or to ISLA 101. The numbers in this first cohort are very, very small. We're looking at 4 individuals, 3 of whom are on ISLA 101, 1 is on placebo. You can imagine the way this could work is that we might have 1 person who has dengue, and then we have 3 people who are on ISLA 101 who don't have dengue, or maybe have reduced symptoms or reduced virus level. These are all the things that we're going to be looking at in this clinical trial. I'm very happy to say we put out an announcement recently giving you an update on the status of this study. If you look at the timeline here, we're actually sitting right now at about day 47 post-dosing. This is post-dosing prior to infecting these folks with the virus. So the first 30 days following the infection is really when the action happens for this study, when people would be exhibiting symptoms associated with an infection, exhibiting viremia in their blood or an increased virus level in their blood. All of those, the first 30-day samples have been shipped off to analytical labs where they're doing the work to crunch, hundreds of samples of blood samples to determine the blood level of ISLA 101 as well as the virus level in these samples. We're compiling these data right now. We'll be sharing them with the Safety Review Committee shortly. The Safety Review Committee is being asked really 2 questions. Is it safe to give ISLA 101 to these individuals? Do you see signs of activity against the dengue virus? If the answer to both of those questions is yes, then they will recommend moving to the phase 2B cohort, which we are scheduled to start dosing in January, which is right around the corner. The phase 2B cohort is now a treatment situation. So we're going to first infect individuals with the dengue virus. Then after about a week, when they should be showing symptoms associated with that infection, they should have detectable virus level in their blood. And we're going to start treating them and ask, can we increase the rate of clearance of the virus? Can we reduce the symptoms associated with that infection? Again, both are very important. Both will provide information about what's our ongoing strategy, what's the next clinical trial that we run. So a very exciting time. We'll be able to talk about the phase 2A results before the end of this year, before Christmas. And then we expect to be dosing the phase 2B cohort beginning in January with data coming out of that probably by around Easter time is what we think. Let's go to the next slide, please. Stephen Thomas, who's on our scientific advisory board, he also is head of infectious diseases at SUNY Upstate, is a big fan of challenge studies, as am I, obviously. And we asked him, Stephen, why would you do a challenge study? And I loved his answer. When there's no available animal models of a disease, and in our case, for instance, non-human primates don't make great animal models. They can be infected. They don't get sick. So it really is important to go to a place where we can study, as Stephen says, looking at a weakened form of the virus, create a mild, safe form of the disease in a well-controlled environment using a small number of people. And that's exactly what we're doing. We have a small number of people in a very tightly controlled study. And as Stephen goes on to say, it's good timing for Island to test their product ISLA 101. Now that we've completed our phase 1 safety study, the phase 2 trial can then examine in a small, very controlled and safe environment if ISLA 101 has impact on viral replication in humans, the symptoms, and the immune response. And those are all the questions that we're asking. We're hoping to have answers for you soon. Next slide, please. This is a slide that has a lot on it, but I want to include it because I think it is very important. This is example of data from one individual that was treated with the same virus that we're using in our current clinical trial, the one we've just completed dosing. And the amount of data that you get here is just robust. So what we're looking at here is days post-infection, and then the level of a variety of factors that we can monitor. But I want to draw your attention to the curved blue trace. This is looking at the virus level in the blood of these individuals. And you can see that by about day 4, the virus level starts to increase. By day 10, it peaks. By day 14, it's gone. That's actually a pretty reproducible curve that we see between many of the people who are infected with the challenge virus that we're using. Some of the other things we can measure, if you look at the red trace with, I can't tell, I think it's a triangle. This is measuring fever. This particular individual had a little bit of fever, not too bad. That's not unusual. Sometimes people have more or less fever. If you look at the blue and the green traces, these are the lines above the fever. These are actually measuring antibody response to the virus. And I'm very interested to see what effect ESL 101 would have on the antibody response to the virus. I think that could be a very important thing for us to learn. Other things we can look at, if you drop below that graph, you see the bars or the boxes. These are symptoms that these individuals present with. Some of these people present with abdominal pain. This particular individual complained about abdominal pain, eye pain, fever, rash, nausea, and vomiting, all very typical of a dengue infection. So with this, what you're seeing now is a person who's not on drug. What we would expect to see, and use your imagination to say, what is it going to look like if somebody has a response to ESL 101? We might expect that viremia curve to reduce or shorten. We want to reduce the area under the curve of viremia. Or if you look at the signs and symptoms, we want to see the number of symptoms be reduced. We want to see the severity of the symptoms be reduced. Any combination of those should be viewed as an effective response where we're reducing the symptoms and signs. So again, these are data that are coming out shortly. We're working on it. We'll keep you across this. Obviously, I think we're all pretty interested to see the results of our study. Next slide, please. Next slide, please. We've been saying for a couple of years now that as much as we love ESL 101, we recognize that it's important, very important for the company to have additional assets for a whole slew of reasons. One, we think we are particularly situated to be able to develop drugs that meet the boxes that we view as important in our strategy. So we've been looking, and we have screened hundreds of compounds that we thought might be useful. The filter through which we look at these compounds is, do they come to us with clinical history? Are they small molecules? Do they have antiviral activity? That's important. Might they be eligible for a priority review voucher, which I'll tell you about in a moment? Might they be useful in a national and military preparedness effort, for instance, countermeasures against nasty viruses? Maybe they're eligible for non-dilutive funding. We think all of this is important, and we've developed a particular set of contacts and relationships that we think these type of assets would be important for us. The priority review voucher program, just to give you a quick intro to that, this is a program that the FDA created to encourage companies to develop drugs for underserved diseases. And there's a few different types of PRVs, is what I'm going to call them. What we're pursuing are the tropical disease PRVs. And to get them, you get your drug first approved in a disease on a list of diseases that Congress and the FDA has established. And things like dengue is on the list of diseases. So ESL 101 is eligible for a PRV. The other assets we're looking at, also, we want them to be eligible for a PRV, because when you get your drug approved, you get a voucher that you can sell. And the most recent one sold, for instance, for $150 million US. So we like the idea that we could have 2 cracks at this. We think that's really important. Let's go to the next slide, please. After all the searching that we've done and the analysis that we've done looking for additional molecules, I'm actually really happy to let you know we've identified one that we like very much. And in fact, we have executed a binding letter of intent with a company called BioCryst. BioCryst is a company based in the United States that had been developing this molecule called galidesivir. It is a small molecule, antiviral, broad-acting antiviral, has activity against some really nasty viruses, quite frankly. The viruses where there is the most data would be Ebola and Marburg. And while these have a significant need for therapies, there's no therapies for these because there are outbreaks in Africa that we would see and you might have heard of. The real reason these become very important is we're of the concern that these can be bioterror molecules, that they may have been weaponized, and we tend to think that that might be the case. So there's significant interest from governments to have countermeasures to be available in the event of a bioterror attack. Galidesivir has great activity. So again, looking through the filter that we look at, does it have phase 1 data? It does. They've completed 2 phase 1 safety studies with excellent results. Do we have activity against different virus? Yes. More than 20 different viruses. This has broad activity, but importantly, BioCryst did some amazing work where they performed non-human primate studies and could see that galidesivir was protective against both Ebola and Marburg infections. So it's a very, very exciting molecule. We're in the middle of diligence right now, but we expect to move forward with this as quickly as we can when we get some answers about the regulatory path. There's some interesting regulatory paths. Potentially the animal rule might be available for us. The animal rule is a program that the FDA created that enables people to get approval without running human efficacy studies when it is unethical or unsafe to run human efficacy studies. And you could imagine that if you're looking at Ebola and you're looking at Marburg, it would be problematic to run a placebo-controlled clinical trial because the placebos would not be well. It would be a big problem. It would be unsafe to run a placebo-controlled study. So we believe that we may be able to run an efficacy study only in animals. And if so, then that could be a really fast path to submitting a new drug application. Next slide, please. The commercial opportunities, I think we can probably all see, are robust. On the one hand, I think we look at ISLA 101 as a potential travelers market. We're looking for people who are traveling to endemic regions. This sort of looks like anti-malarials. For those of you who are familiar with taking anti-malarials before you travel to an endemic region, the anti-malarial market is approaching $1 billion market opportunity, US dollars, about $1 billion. So we think that that in and of itself is a significant market. But we also are aware that the military is interested in what we're doing with both of the programs we're talking about. We think there's a significant need to stockpile potentially ISLA 101 as well as galidesivir for national outbreaks. And then again, both of these molecules are eligible today for a priority review voucher. Next slide, please. Corporate snapshot. I won't go through all of this in great detail. I think our stock price maybe closed yesterday at 16.5 cents, so down a little bit from when this slide was made. We've had a really nice run recently. I think in large part due to the financing that we were able to recently close. It's really set us up nicely, and we're in a position now to be able to drive through this phase 2 study, get those results, as well as execute on the galidesivir program. Next slide, please. The key milestones, I'm not going to remind you of what we've done over the last year. What I really want to say is we're in not just the middle of the first cohort of the phase 2 study. We're nearing the end of this study. We're getting close to the point at which we can see these results and that we'll be able to talk. We'll talk to you about them. We secured financing, $3.5 million, which again is going to support us for about the next 15 months and allow us to do a lot of very exciting things. We're expecting the data readout from the phase 2A cohort in short order within the coming weeks. We're continuing our due diligence with the galidesivir program, a program we like very much. And of course, throughout all of this time, and I think I've mentioned it before, talking to potential partners. So I frequently find myself in conversations with pharmaceutical companies in Brazil or in Singapore, letting them know what we're doing. I think that these are companies in countries where they have a significant dengue problem, and they're very interested in what we're doing. Starting in January, we plan to dose the phase 2B cohort, the therapeutic cohort of the challenge study that we'll be doing with a data readout probably by around April. We'll then be conducting an end of phase 2 meeting with the FDA to talk about what our next clinical trial looks like. So again, the next weeks are going to be exciting. The next 6 months are going to be exciting. I think it's a great time to be looking at ILA Pharmaceuticals. And again, I want to thank everyone for their support. I think that's my last slide. I think I've got to thank you on the next one. So I'm going to close that. And thank you all again for being here. Thank you for those of you who are online, and I'll be happy to take any questions.